to the edge. Okay, so you're gonna get this quadrant right here, the one with the bigger squares, and then you look at this quadrant here. So basically the four corners. So you have four quadrants, and so you can name them 1, 2, 3, and 4. And then the volume that fits into these quadrants is already been determined. And so the volume here is 10 to the negative 4 mLs. Okay, so this is the volume that fits in each one of these squares or quadrants. Okay, so when you put your cells here, when you're trying to get the you had to estimate the number of cells, you're always going to multiply the cells by the reciprocal of this number, which in this case is 10 to the 4 or 10,000. So whatever number you count in this grid, you're going to multiply, at least if you haven't diluted the cells, you're going to multiply by 10,000. Okay, and so let's say you have 10 microliters and you don't dilute the cells, but then you count the cells. And let's say you just count this quadrant, and let's say you have 4 cells here. Okay, so 4 cells times 10,000, it gives you 4 times 10 to the 4th, right? And so this is how many cells. Right. This is how many cells you're going to have in the sample that you put in there. Listen to the 4 cells per amount that you put in there, but it per ml actually, because you're using these concentrations. So you're converting the dilution. It's like a dilution here, but this is the amount that you have here. If you only have, let's say you only put a few microliters here, then you will say 4 cells in 4 microliters. But because this is, you usually report it per ml, so you always have to multiply by this by 10,000. Okay, so this is one of the first things. So the cells are going to land here. And the next thing is, in order to see the cells, you had to stain them. And you can see living cells in the in this grid, in this slide, but you can also see dead cells, and you don't know which cells are dead and which cells are living. And so what you do is you add these stain. So normally to see cells, you had to stain them. And so here they use this stain called trypan blue. In this example, what I did is I took 50 microliters of trypan blue, and then 50 microliters of your cell culture. So in this case, we're diluting 1 to 2. So whatever number you get, you multiply times 2, because this is the dilution factor. This is how you diluted it. So what happens with trypan blue is that trypan blue stains cells that are dead. Okay, so trypan blue will stain dead cells. And this is because the membrane of dead cells, it's the membrane of the dead cells is not stable anymore. In cells that are viable, so this is trypan blue, cells that are viable or living are going to be yellow. So dead cells are blue, and living cells, or what people call viable cells. So viable cells are cells that are going to divide later. They're called viable cells. So these are stained yellow. Okay, so the reason to stain them is because normally you only care about viable cells. You know that dead cells are not going to replicate, And you also want to see if your culture is healthy. If you have a lot of dead cells, then your cell culture is not very healthy. Okay, so this is what normally what you're going for, viable cells or living cells. Right. So this is a depiction of something similar what you will see under the microscope. So I had this grid here. So remember, this is a grid, and you're going to count the four squares, 1, 2, 3, and 4. And what you're going to do is you're going to if you want the number of viable cells or living cells, you're going to count each square. Okay, so let's start with the living cells first. Okay, so living cells, remember I told you living cells are yellow. Okay, and we have four quadrants. We have 1, 2, 3, and 4. Okay, so in quadrant 1, so you want to count the cells that are inside this quadrant only. And some people have these, sorry, I erased that. So some people have the idea that you don't count all the cells that land like between the edges. So like a cell here, they may or may not count it. So they have this system, and so where they only count, let's say, the edges that are on the outside. So you can count the edge, if any cells that falls on the edges, you count them, but the ones that fall inside the edges, inside the slide, you don't count them. So they may count these, or in this case, they count these and these. And so I like to count the ones that are on the outside. Okay, so this is, you're gonna count the cells in the inside too, but let's say you have a cell that lands here. Okay, so because I'm only counting the outside, I'm not going to count this one right here. But if I have a cell that lands here on the edge, I'm going to count. But regardless, if you don't have any cells on the edges, then all you do is count the cells inside. Okay, so the living cells, so like I said again, living cells, so we're going to count living cells. So we have four quadrants, 1, 2, 3, and 4. Okay, so quadrant 1, we count the living cells, the yellow cells. So it's 2, 4, 6, 8, 10, 11. So here we have 11 cells. And then you go to quadrant 2, and then here you have 2, 4, 6, 8, 9 cells. And then you go to quadrant 3 here, and you have 2, 4, 6, 8 cells. And then you go here, and you have 2, 4, 6, 8. Okay, so I lost count. 2, 4, 6, 8, 10, 12. So you have 12 cells, quadrant 4. Okay, so you're going to add all the cells. And here, I think I have 40. So 40 total cells, and then you divide by the 4 quadrants. So 4, and so basically you have an average of 10 cells per square. Okay. See, so what you're going after the average. Okay. So they use the average of 4 quadrants because sometimes you may have 4 cells here, and you can have 12 cells here, and then here you may have 14 cells, and here you may have 7. And so none of these are close to each other, 14 and 12, 14 and 4. And so you take the average. And so that's more accurate than just counting in one well, because you don't know where the cells are going to land. In any cell that falls outside of these quadrants, so let's say that the cells land here, you don't count them. So you don't count those. You only count in here. And because otherwise you have to multiply by a bigger number. Okay, so now you calculate the average cell number so far. So now let's say what will be the number of cells if you have a cell culture and then you dilute it by 1/2. So you dilute it by 1/2. And so the way you calculate it, let's say I'm using the example that I just went over, where we have 10 cells per square. So 10 cells per square. So this is how much you count it. Okay, so you want to know how many cells you have in your sample, then the answer is going to be 10. The cells that you counted, and then you're diluting by 2. So here the dilution, the diluting by 2, you write the reciprocal, right? And then remember I told you, you always multiply by 10,000 because you want the cells in mLs. Even though it doesn't say here per ml, so just assume that it always going to be mLs. And so you multiply by 10,000, and so the number here is going to be 2, my zeros are correct, 2 times 10 to the 5th. Okay, so that will be cells. This is going to be per ml. And again, these are viable cells only. It's not asking you about dead cells. So these are viable cells. So these will be your answer. Okay, so in a quiz, if you want to write this number, you don't have to write all the zeros. You can just say 2 exponent to the 5th. I know you meant this. Okay, then you write cells per ml. That will be your answer. So that way you don't have to write these 10 to the 5th, to the 5th power. Okay, so any questions so far? Is it clear? Okay, so now I can ask you also how many dead cells you have. So in terms of dead cells, and so when you do a cell culture, you actually have to do it within 20, 30 minutes, otherwise the cells start dying. And so the dead cells are all blue. So we do want to count dead cells. Maybe I should use the blue color. Dead cells is the same thing. So dead cells, you're going to count 2 here. So 2. Then I made my life easier. I put 2 here too. And then here I have 2. And then here I have 3. So I don't have a lot of dead cells, but I do have some. And so when you add these, this is 9, right? Now 2, 4, 6, 3, 9. When you divide it by 4, that gives you 2.25 cells. Okay, so these are the dead cells. So again, this one depends on the dilution. If you dilute it by 2 again, then you multiply times 2, and then you multiply times 10,000. Okay, so in reality, it doesn't seem like you have a lot of dead cells, but we do. We have, so 2.25 times 2 is 4.5, right? So we have 4.5 times 10 to the 4 dead cells per ml. Okay, so now, so these are dead cells. So now you wanted to know what is the fraction, what is the percent of living cells, percent of viable cells. What you want to do is you want to you get the total number of cells, living or dead, but percent viable is going to be viable cells, right, over total number of cells. Total number. The total number of cells. So in this case, the viable cells we counted was how many? 40? 40 viable cells. And then the total number of cells is going to be 40 plus 9 dead cells. That's 49. So the total number of cells is 49. Okay, so when you get the fraction, so it's going to be 40 over 49, I think it's like 0.8 times 100. That's going to give you like around 82% of viable cells. Okay, so it kind of tells you that your cell culture is healthy. You don't have a lot of dead cells, or percent wise anyway. So this would be the fraction of viable cells. Okay, not the total, but the fraction percent viable. Okay, any questions? Okay, so again, this is the calculation that I did previously. You take the average number of cells. You always include all the dilution factors that you use, all the dilutions that you did. So in the first one, the dilution factor was 2 because I diluted by 2. And then the cell suspension, the conversion to mLs. So this is how you get that.
I do have one question because I noticed like for the dead cells,
yeah,
we had 2.25 cells. So we can't really have like 0.25 of a cell. So like in that case, like, or can we, I guess is my question, just because like 0.25 of a cell per each square, wouldn't we need to round that up?
Oh, you can round it up, but that just means so that means that it's an estimate, right? But a very close estimate. And
Right.
so yeah, so the numbers I did is that's I just made up with those numbers. That's where you have a fraction. But yeah, you would always have a fraction. And so yeah, that doesn't mean that you have 2.25 cells. No, it just means in average, right? So the average. So if you have, let's say you come up with 2.5 or 4.5, 2. What was it? 2.25. Yeah, 2.25. Yeah, that doesn't mean that you have that's the 2.5 cells instead to the 4 or 5 per ml. No, that's just the average. The average could be, I don't know, depending on how accurate it could be anywhere from, let's say, from 2 to 3. Right, so it just tells you it's probably closer to 2. But yeah, you can round up.
Okay. It's just because like I don't know, like I think of it and I'm like, okay, so we're taking the total and then we're dividing by the 4 quadrants. So when we do that, you get the average, the cell average, and you said that's what we're looking for. But when you look at it at this the number, the value 2.25 cells, we can't. It would be an inaccurate statement to say two. Right? So wouldn't we have to, like that's what I or I don't know. That's why I'm asking.
No. Okay, so remember we have power of 10. So
right.
In reality, you're really counting how many zeros do we have? 5. So if it were only a few cells, yeah, that would be weird, right? But in reality, we have 5. Let me see, 1, 2, 3, 4, 5. So in reality, the number is this. So this is a real number, right? So the way to report it, yeah, it's 2.5 times 10, right? Well, here is not 2.5, but the dead cells was 2.5. So
Yeah,
now,
it was
yeah,
2.5 times 2, right? Because we diluted the cell culture. So
yeah,
then once we diluted that, then we multiplied by 10 to the 4,
right,
10,
yeah.
10 to
So
the 4th.
yeah, so here you don't need to so there's no decimal here. So it's
Right.
not 2.7, it's two, what is it? 250,000, right? So yeah, 200 is a whole number. So yeah, you report the whole thing. So
Okay.
in this case, yeah, you report. You don't have to round up. Even even if you had 2.1, yeah, you don't. You report it as 2.1 because you're basically saying it's 200,000, 210,000, right? Because if you win the lottery and they offer you 200,000 or 220,000, you're gonna pick the 220,000, right? Because it is a bigger number. So it's the same thing here. It is a whole number. So yeah, but if you only have a few cells, yeah, it's not. You don't have 2.5 cells. That's right. Okay, so now let's have another example. So now let's say you have a question where it says you have a plate and you want to know what is the number of viable cells. And you do two, in here you're doing two dilutions. You dilute your culture 1/4. So the first dilution, you diluted 1/4. But then you realize that, so let's say these cells are in a test tube, you had too many cells here. So you diluted again, and this time you diluted 1/10. Okay, so you went to 1/4 and then you diluted to 1/10. Okay, so now, so this is doing a 1/10 dilutions. Okay, so what is the number? And let's say you have 10 cells, average of 10 cells per square using the same number as before. And so, can anybody tell me how do I get the answer here? What will be the answer? How do I come up with the answer here? What is the first number I put here? So everything is given to you already.
So you will put 10, right?
Yeah, you put 10, right?
And then you will put 4 because it's the reciprocal of 1/4.
Correct. So 10 times 4.
Would you also put another 10 because it's dilution with 1/10 also?
Correct.
No.
Yeah, you have to put 1/10 because you diluted twice. Very good.
And then after that, it's just cell suspension, so 10,000.
Yeah, 10,000. Yeah, very good. Correct. So that's exactly how you do it. Okay, so uh, sometimes I forget my zeros. I mean, 1, 2, 3, yeah, 10,000. Okay, so the answer here is going to be 4.0 times 10 to the 1. Let me see how many zeros do I have. 1, 2, 3, 4, 5, 6. Is it 4 times 10 to the 6?
Yes.
Okay, so that will be the answer here. Cells per ml. Okay, good. So yeah, so now you know. So now you know how to do dilutions. Okay, so I was going over this because you're gonna have something. You get to know this for your quiz. And, okay, so yeah, I explained it here. I made a mistake here. So as you can tell, I put 2, but it should be 4, right? So 4. Okay, so here it explains this is the average number of cells, viable cells 10, which is here. Then times the first dilution is 4, which is 1/4. The second dilution is over here, is 1/10, which is right here. And then you take into account the amount that you put in the cell in the slide. So this is 10 times 10,000. Okay, so this is how you do it. Okay, so if you have any questions, let me know. If not, I'm gonna post a video. It's the same thing that I went over here right now. It's a voice thread. It's a video. It's gonna go over this again. You don't have to watch it. It's completely optional. You don't get any points for it. It's just in case you miss something or want to go over it again and you want to hear somebody explain it to you again, you can watch the voice clip. I haven't posted it yet. I'm going to do that right after class. Yeah, I posted it for my Monday class, but not for you guys. I wanted to go over it first. Okay, so this is, any questions about delusions? He was a calendar, is it clear?
Yes, very clear. Thank you.
Okay, good. Okay, so if after class, right now, when I'm done, if you have any questions, you can ask me, or again, you can watch the video before you take your quiz. And so now I'm going to go to the next presentation, which is what we were supposed to do today if we were in class, but I'm going to go over the background. Okay, so I just heard that we are not going back to school until maybe after spring break. Okay, so I'm hoping we'll at least be able to do a few experiments when we are back. Okay, so but now the topic is different. So now this is affinity chromatography. And has anybody here ever done affinity chromatography?
Professor, you're supposed to be sharing the slide?
Yeah, I'm just,
Oh, it's because I don't see them.
can you see it now?
Yes, sir. Thank
Okay,
you.
so this is affinity chromatography. Has anybody ever done chromatography or affinity chromatography?
Not at all.
Not at all. Okay, that's fine. That's great. So affinity chromatography is basically you have a protein, and then you have a ligand that binds to your protein, and you want to purify your protein, right? So you have a ligand, it sticks to your protein, and you can use that interaction to purify your protein. And so what happens is, let's say we look at the blood. So when you look at a sample of blood, let's say you have a blood in here. How many proteins does the blood have? Or let's say not even blood, but the serum or the sera. What kind of proteins that the blood has? So you're looking at blood or serum. Let's say blood, or better serum, because we don't use the blood normally, right? So you have serum samples. What kind of proteins will you have in serum? Anybody remember?
Albumin.
Very good. The major one is albumin. Excuse me. This is my last class today, so my voice, I'm kind of losing my voice. So yeah, so you have albumin. So this is in very large quantities, right? What other proteins are there in the serum?
Globulin.
The globulins, right? Yeah, the globulins, and there is more than one, right?
There's 5, right?
I'm sorry?
There's 5, right? The IGs.
There are other proteins, IgGs, the different IgGs, globulins. There is a protein transport proteins. One is called ferrodoxin. Let's see if I spell it right. Ferrodoxin. There's ferrodoxin. I think this is it. Okay, so ferrodoxin, and then there you have fibrinogen. In the IgGs, all kinds of fibrinogen is involving clotting. So anyway, so that the point here is that in serum, you have you have all these types of proteins. And not only proteins, you have sugars, you have salts, you have a whole lot of things inside the sera or the serum. And so let's say you want to purify from this soup, you want to purify just one protein. And so let's say you want to purify IgG. Okay, so you don't want IgA, so you don't want albumin, you don't want IgA, you don't want You don't want IG, what are the other IGs? IgG or any others? You just want this one, IgG. Okay, so in order to purify that one specifically, then you have to find a ligand that binds to it only to that one and not to the other ones. That ignores, it has no affinity for the other ones. This is why it's called affinity, because the ligand has affinity to only one compound. It can have different affinity levels, but it has only one strong, very strong affinity. So what happens is you have a protein. So remember the proteins are three-dimensional structures, and then let's say my protein IgG looks like this, just for it's an example. And so in order to purify this IgG, I had to find something that binds to it specifically, right? It doesn't bind to albumin, it doesn't bind anything else. And so that's something that binds to it. If it's an IgG, is going to be like an antigen, right? So it turns out this not protein, but whatever binds to this, the one you want to purify, this is called the ligand. Okay, so it can be like an epitope of a protein. It can be a protein. And so there is a molecule called protein A. Protein A is from Staph aureus bacteria. And so protein A, it's made by bacteria on the surface, and it happens to bind IgG. Because so the ligand for this interaction is going to be protein A. And so what you do is they make a chemical reaction so that the protein A binds to a bead. Okay, so you have protein A, and let's say protein A looks like this. Okay, so protein A, and it fits, it's able to bind to the, because it has the right shape to bind to these IgG. So this is going to be protein. And so the protein A is linked to a bead, and the bead is like a sphere. The bead is it's called a matrix, and this bead is going to be huge compared to these molecules. So I'm only going to put a little bit of the bead surface here, and so you're going to have these spheres made. It's a synthetic. You don't need to know what it's made out of, but it's going to have all these ligands bind to it. Okay, so all of them covered with protein A. So I'm going to write this like a banana. And so this is going to be protein A, and its link is it's linked with in a way that it's not going to come off out of the beads. It's a very stable chemical reaction, so it doesn't come out. The protein A doesn't come out of the bead. Okay, so you have protein A, and so remember, this protein A is the ligand. Okay, so what happens is when you put these beads in a column, so the column is made out of plastic, or it could be made out of glass. When you put all the serum through these beads, you're going to have different proteins, right? You're going to have, let's say, you have albumin floating around here. So this is albumin. The albumin is not going to bind here because it's not the right ligand. It doesn't bind to it. You're going to have other IgGs. Okay, so let's say one IgG here, and this different IgG, but they're not IgG. I mean, a different immunoglobulin. And then let's say you have ferredoxin here. So these squares represent different proteins. None of these are going to bind to the ligand. None of these, because they don't interact with it. Only the IgG. Okay, so all these proteins can flow through them and don't bind to them. And so the IgG is going to stick to the only. Okay, so then you wash the excess proteins. And so what you have at the end is all these other proteins are gone, but you have your IgG. So your IgG molecules like these, they're bound to your beads. Okay, so you have everything else is washed off, and you have U protein bind to the beads to the protein A interact. And so all you do now is you change the pH, or you add high salt. And what the salt does is that you have interactions between two molecules. The salt breaks those interactions. And so you have the ligand and your protein bound like this. You add very high salt, like sodium chloride, or even detergent, or a competitor that competes with the binding. And then so this, your protein IgG is going to come off from the protein A or the ligand, and then you can wash it, and then it will go to a tube that you're going to pick up your purified protein. Okay, so this is a really very useful technique that is used all the time to purify protein. The only thing that you have to do is you have to have a ligand. A ligand has some kind of molecule has to bind to your protein that you want to purify. And so those are the basics of affinity chromatography. Okay, so some of the things that they say about affinity chromatography, what it does is that you can actually pick a small molecule from a soup that has thousands and thousands of proteins. So it has a capacity for high capacity to purify the protein that you want to. It is very selective as well. And the other thing it's going to do is going to concentrate the protein, because you elute your protein in this very small amount of buffer, and so the protein that comes out of your column is can be highly concentrated. And the other main thing is you get a high amounts of protein. Okay, so you can purify a protein that was only a few microliters. A few concentration is low per ml. You can purify it to like milligrams per ml. Okay, so this is a very good, very, very useful technique, and it is done in a lot of protein labs. And so this is just shows you, this is describing here what basically the steps that you do. So in this case, they use bacteria to make protein. So you clone your protein, the gene of interest that makes your bacteria, you clone it in E. coli, and E. coli makes an excess amount of your protein. You stimulate your bacteria to make your protein. And then so you lyse your bacteria, you break it. So this is bacteria that has your protein in there, and then you lyse it. And then you have your protein in solution, but then you have a lot of other proteins from the bacteria. So you have a mixture, again, a soup. And so what you do is you pass this soup containing you through a column like this, and then you can purify your protein at the end. So it's basically the same thing that I did, that I explained to you. And then you can determine how pure your protein is doing the an agarose gel called the SDS agarose gel that I told you before. And I'll show you one later. So SDS page, acrylamide gel. Okay, so what happens is you can monitor how your protein is coming, where it is in the column. And so again, so you're going to have the columns for affinity chromatography. It usually is small. It's a piece of plastic about this size, a column about this size, and so it has so you have these. It looks like a tube here, and it has a filter here at the bottom. And so what you do is the matrix, what is called the matrix or the beads, there you put them in here. Okay, so these are the matrix that has your ligand. So these are the beads, they're tiny, tiny little beads, and it has linked to your ligand. So in our case, the ligand, like I said, is protein A. It's a protein A. And so what happens is you're gonna put serum. So the serum goes here. So animal serum. In our case, we use, I think, rabbit and I think horse and something else. So you put the serum, and all the proteins that don't bind to the column are going to come out here. Okay, so different proteins. So I'm going to use different colors for different proteins. So all the proteins come out. So if in the case of the serum, the albumin is going to come out in really, really big amounts. So let's say this is albumin, so you have a lot of albumin. Okay, and your protein that you're interested in is going to be stuck here. So the IgG protein is going to be stuck here. Okay. Okay, so if you have all your proteins is here and everything else is out, you might lose a little bit of your protein, but for the most part, your protein is going to stuck to the column. And so this is what they're showing you here. They're showing you the beads. So this is a bead with protein A or the ligand. So the ligand, and then you add the serum. So all of these colors represent the serum. So and then the serum comes out, the serum proteins, and your protein stays there. So this is what they show you here. Your protein of interest stays attached to the beads. And then you wash this, and then your protein comes out. And what they show you here in this graph is that you can follow using using a device that measures absorbance. And so if you remember from biochemistry, the proteins have amino acids, they have so much absorbance, right? So something like phenylalanine. So any protein that has these aromatic compounds, at least one or two tyrosine, I think, they can measure the absorbance, and so you can track where the proteins are coming out, because what you do is you have a column, and then you have different test tubes where you pick the different fractions. Okay, so the washes you pick, and so if you use a machine that measures the absorbance, in the beginning all you do is get a flat absorbance. That means there's no proteins here. And so this is the volume. So let's say you wash in here, but then you add your sample, and then you see a really big spike in absorbance. That means there's a lot of protein. Okay, so this is the proteins that are coming out. These are these proteins. These are the proteins you don't want. So these are you can think of it as protein waste, because you don't want. These are the other proteins that were in solution that you don't want. Okay. And it has a big, so it has a big absorption here, large amounts of absorption, but it also comes out at a very large volume. This is because there's a big amount of protein in here. So this is volume. And so you keep washing. So this is when you start washing your column. So once your waste comes out here with all the proteins you don't want, then you wash with buffer. Okay, so you wash and wash and wash. You wash with buffer, and your protein is still going to get stuck here. It's still attached to the ligand. And so eventually, so you wash with buffer until it gets to 0. So there's no more protein in here other than you coming out of here other than the ones that's stuck there. And so then you add your salt here, somewhere in here, or something that's going to break the bond between the ligand and your protein. And then these are going to dissociate. And what you do is that you get an increase in absorption, and this is the protein that you want. This is going to be your IgG. So this is your IgG, and this is what you want. And so one thing you do see here is that the volume is small. And that is because your protein is very concentrated. Okay, so those are the basics of affinity chromatography. Okay, so some of the things, so once your protein so now you have your column here. You have your column, and you had the filter here, and your protein, it's inside the matrix here. So you have this matrix, and your protein is stuck to the ligand in here. So let's say these circles is your protein, IgG. Okay, so it's stuck there to the ligand. So you have IgG bound to protein A. So I'm just gonna write for sure protein A. Okay, so you have all your IgGs are bound to protein A. Okay, so now to break this bond here, to break it, then you can change the ionic strength. So that means you can add salt, some type of salt. You can add, there's a compound that breaks, it's called imidazole. You don't need to know it, but it changes the ionic strength, similar to salt. You can change the pH, or you can add a ligand that binds to your protein or to the protein A better than your protein. And so all of these will disassociate, and then your protein by itself is going to come out of the other end, pure. Okay, so you can also add detergent. Okay, so some of the requirements for affinity chromatography, I already mentioned, that you require a ligand that it has a covalent bound to the bead or to the chromatography. So when I say bead, I really mean the matrix chromatography. That's the name that is given to chromatography matrix. But again, these are spheres made out of a compound that doesn't react with proteins. And then you link your ligand to the beads or to the matrix. It's called a matrix. Okay. Okay, the other thing you need to do is you need to be able to break the bond between your protein and the ligand. And so in this case, if the bond, if they bind too tightly and the binding is not reversible, and that there's a few proteins that binds ligands irreversibly, then it's not going to work. So you have to be able to break this bond. Okay, so here are, in our case, protein A is the ligand. But there's some other things that are used, like antibody antigen interaction. So you can think of protein A as an antigen. So this will be protein A is the antigen for the IgG. So that will be our example. You can have an enzyme. This the substrate, you can have something called metal ion. So there's some proteins that are, you can think of a protein as being linear just for our purposes. Usually they're not linear. And what you can do is you can add these amino acids called histidine. Histidine. You add like 6 at one end. And so what happens is you're tagging, you don't have a ligand, but you're creating a ligand by adding these histidines. You clone it with your bacteria when you clone your gene. And so you have your protein here with these histidines. And there is something called there's a column, it's called the zinc column that will bind the histidines specifically. Okay, so it's called the histag proteins. They're tagged with a hist. And so your protein that has these histidines will bind to this column here that it's called the nickel column. It has the compound nickel that preferentially binds very well to anything that has histidines. And so you have an excess of histidines, so it will bind to your protein. Okay, so those are some of the ways you can okay, so I'm getting to the end here. So in our case, so if we go to the labs, well, we're not going to go to the lab soon, but if we're going to go, we might be able to do this where we have a column that is already linked to protein A, so that would be the ligand. And like I said, the protein A is from Staph aureus, and it binds specifically to IgGs. Not going to bind to anything. It has a different binding affinity, though, for IgGs from different species. So an IgG from a rabbit may bind different than an IgG from a human. Okay, and so this is one way to purify the IgG. And so the rabbit and the human actually will bind to protein A really better than, let's say, the horse. Okay, so one thing about this protein A column, so we already have this in the lab. So we have already have these columns that we made last year. It has the column. We try to make a better drawing. So we have columns. They're made out of plastic, and they had the beads already here. And these beads already had the protein A inside here. So we had the beads here, and all we had to do is add the serum, the animal serums or the serum, and then we collect different fractions using test tubes. So in a test tube, you can collect whatever flows into here, whatever comes out, you collect it, and then you put another test tube, and then you add buffer to wash everything that is in there. To add buffer, and you keep adding buffer, and so you collect that too. So you collect that. So this is called the wash. In here, your protein is already stuck to the column in these steps. And then after the buffer, you add something to break the bond here, okay, like a salt or a change in pH. And so let's say we add salt, and then you get another test tube that's going to have your IgG. And then what you do, you take all these samples, 1, 2, 3, 4, 5, and then you run a agar SDS gel, and you put all your samples in the gels, and then you're gonna get something like, let's say the first one that just has the flow through. You're gonna have a lot of albumin here at the bottom. And then you're gonna have all the proteins too. And then as you wash, the amount of albumin goes lower, and then then eventually your purified protein is gonna be over here. But the other bands are gonna be gone. And so if it's pure, you're gonna have only one band. And then you have to test this band to make sure it's IgG. And to do that, you can do an ELISA. Okay, so that's what this lab is supposed to be about. Okay, so one thing I was telling you is the IgGs bind with different affinities to the protein A. So protein A binds better to some immunoglobulins than to others. So in here, it has all the Ig immunoglobulins from humans. And here you can see there's two types of IgG 1 and 2. And so it binds really well. So 4 classes means that it binds really well. But on the other hand, if you take the IgG of the mouse, you can see it doesn't bind as well. So using this procedure, you won't be able to purify IgG from mouse because it doesn't bind really well. And also from rats. So the IgG from rats doesn't bind as well, or from sheep. And so last year when we did this, I believe we used rabbit. So here, rabbit, we use rabbit, and it bound very well. And as a control, we use sheep. Sheep supposedly is not going to bind as well. And we use cow too, I think. That one doesn't bind. It still binds, but not as well. Okay, so the procedure to do this is here, but I'm not going to go over this because this is only if you were doing the experiment. But what I'm going to mention though is that this is how you do the experiment, and the salt that we use to elute your protein is 3 molar magnesium chloride. Okay, so you can think 3 molar is a very high concentration. So so you go through all these steps, you can go over these. If we get to do this experiment, then we can go over these steps. But basically, you're gonna have a column, and at this step, after you elute it, you're gonna have your IgGs here. And to break the IgG bound to the protein A, we add 3 molar magnesium chloride. I want to say, yeah, 3 molar. Okay, so any questions? So I'm hoping that we go back to the lab, we get to purify some IgG in one of these animals that we have. So I'll go over this later. So these are the examples of results that we got last year. So this is rabbit IgG purified. And so I'll go over these. Also, I did this myself. So I purified this myself. Okay, so are there any questions right now?
No, thank you.
No, did it make sense?
Yes, sir.
Yeah, okay. Okay, so then that's it for right now for me. But again, I'm gonna post the quiz in a few minutes. You just give me about 10, and you can take it anytime today. You can take it anytime tomorrow. And the other thing I'm gonna post is the video on the hemocytometer. If anybody wants to look at it, you don't have to at all. It's optional, completely optional. Okay, so if you don't have any other questions, then we are done for right now. Then so just let me know how it goes with the quiz. You don't need the browser for the quiz. And oh, the other thing is the quiz, once you start, you have to finish it.
Okay, and sorry, I have a question. Do we have to take the quiz right after class? You said that it would be available for us today and tomorrow,
Yeah,
correct?
you can take it anytime until tomorrow midnight.
Okay. And just for clarification on the quiz will cover the past, will it cover the 2 concepts that we covered today, or it's covering concepts 1 through 5 the 5 other PowerPoints?
No. The quiz only covers the dilutions and dilutions factors, titers, just like it says on the, I should say it on the schedule. But anyway, no, it only covers dilutions and because remember we were supposed to have this quiz back before. No,
Yes.
Yeah. So this is covering dilutions. So that's why I went over the hemocytometer, all the concepts of related to serum and blood.
Okay. Thank you.
You know what is blood, what is serum, what is plasma, how do you do dilutions, what kind of proteins you get in in blood. Okay, just give me a minute and I'll let you know what's in the.
Gotcha.
Just hold on one couple of minutes. Maybe I don't remember everything. Okay, I'm back. So it's only dilutions and titers. The quiz.
Okay.
Okay, any other questions?
I think that's it. So since you said it's only dilutions and titers, we don't have to worry about well, we should still know them, but the serum concepts, what's in serum, the specific components of them, we don't need to have them specifically memorized for the quiz because that's not on the quiz.
Right, yeah. So you basically need to understand all the concepts of the dilutions and titers, understand them, be able to explain them, and be able to do the calculations. So you remember the serial dilutions and also the how to count the cells using a hemocytometer. That's basically what the quiz is about.
Okay.
Does it make sense?
Yes, thank you.
Okay. Is there any other questions? If not, we're done. And just remember, you're going to take the quiz in a few minutes. It should be available in 15 minutes. I just had to post it.
Thank you, Professor.
Thank
Thank
you.
you.
Bye-bye. See you next
Thank
week.
you.
See you next week, Professor.
Thank
See you.
you.
Thank you.
Thank you.
Bye.